Nice to
around
see
15
you.
years.
I'm glad that I'm able to meet you guys.
Thank you, Mr. Thanks for your kind concern. Just a moment, sir.
Okay, sir. Not a problem. Take your time.
Thank you, sir.
Right.
Sir, shall we start, sir?
Hi, yes, ma'am. Sir is ready. If our participants are ready, we can start.
Okay, sir.
I think many of you have joined.
Good afternoon, one and all gathered here. On behalf of the organizing committee, I wish to thank you all for the presence for the Industrial Skill Enrichment Program on extraction, isolation, and screening of biologically active phytochemicals. It is possible to move everything through God by prayer alone. Let us all arise and seek the Almighty's blessings to the prayer song.
Praise on you.
Thank you all. To continue with this, it is my pleasure to invite Dr. Meera Parthasarathi, Assistant Professor, Department of Chemistry, to welcome the gatherings.
A very good evening to everybody. On behalf of the management of Srimad Andavan Arts and Science College, respected Secretary, Director, Principal, Vice Principal, Deans and Head of the Department, I would like to welcome the speaker, Dr. Sivanesan Dhandayuthapani, for sparing his valuable time and knowledge to empower us and all the faculty and student delegates for their enthusiastic presence in this beautiful evening. Now we move on to the Industrial Skill Enrichment Program on extraction, isolation and screening of biologically active phytochemicals organized by the Department of Chemistry. Now I hand over the session to the convener for conducting the program further. Thank you.
Thank you, ma'am. It is both honor and privilege to introduce our resource person in this occasion. I call upon Dr. A. Anandan, Head, Department of Chemistry, to introduce our resource person.
Acharya Thiruvanaikoil Potri. Very good afternoon to all. On behalf of our management, secretary, director, principal, vice principal, deans of other departments, heads, faculty of our department and other departments, on my own behalf, I am very happy to introduce our resource person of today's event, Industrial Skill Enrichment Program on extraction, isolation, and screening of biologically active phytochemicals. First, I would like to thank Dr. Sivanesan Dhandayuthapani for his kind consent. I am very happy to introduce our resource person. I have a cordial relationship with him for the past 17 years. He has a lot of academic and research records. He is presently Deputy Dean Research, Santosh deemed to be University, Ghaziabad, Delhi NCR. Our resource person is basically a biochemist, graduated from Tamil University, Thanjavur, Tamil Nadu. He started his career as lecturer who taught for both undergraduate and postgraduate students and mentoring for many MPhil research scholars. Later in the year 2010, he moved to United States of America in view of getting advancement in the field of cancer and natural products research at Nova Southeastern University at Florida and Ohio State University, Ohio. Further, with his expertise, Dr. Sivanesan advanced himself as a research scientist in cancer and natural product research. He has published more than 25 research articles in reputed journals of both national and international levels. He has also presented more than 50 research presentations in various national and international seminars and conferences. With his biochemistry background, he established himself into the field of cancer research to study the advancement in identification of the cancer biomarkers and drug discovery. There are 6 criteria in selection of a resource person: intelligence, successful experience, education and training, availability, ability to communicate, and open-mindedness. I assure you that our resource person fulfill all your knowledge needs. Now, I invite our beloved resource person, Dr. Sivanesan Dhandayuthapani, to share his views on extraction, isolation, and screening of biologically active phytochemicals in this industrial skill enrichment program. Welcome, sir. I am very happy to meet you after a long time. May this session will be an eye-opening session for all our participants. Welcome, sir, once again.
Of course. Thank you so much for your warm welcoming.
Thank you, sir.
I'm also so surprised after so many years I'm at least able to see you online. Thank you so much. Also, I would like to share my view of thanks to the management and also the respected secretary of Srimad Andavan College of Arts and Science, the director, the principal, the vice principal, all the dean of different faculty, and my fellow friends there, also the students who are very enthused to attend this session today. Once again, I would like to express my thanks to everybody to invite me to give this special opportunity to share my knowledge on whatever I acquired throughout my career for the past 20 years with the natural product research, as well as few about my cancer research as well, where I have applied. I extracted the product from plant material and I applied where it was published. I would like to share that also. Good afternoon. Thank you, one and all. I think we can go into the session. Am I audible to you? Hello,
Yes, sir.
everyone.
Yes,
Am I
sir.
audible?
Yes, you're audible, sir.
Okay. Is my screen is shared?
Not yet, sir.
Okay. One second, sorry, there is some glitches. I apologize for the inconvenience. I'm fixing it. Is it visible now?
Yes, sir.
Okay.
Yes, sir.
Yes, sir, clear. Kindly present in full view mode, sir.
Yeah. I think it is in full view now. Is it in full view mode now?
No, sir.
No, sir, no, sir.
No, sir.
Slide presentations are.
Right.
Sir, sometimes this problem arises, sir. No issues. You please continue. We are trying, but sometimes we are unable to find full view mode. It's not a big issue. You please carry on. Or you use entire screen.
Is it clear now?
Yes, sir.
Is it full now?
No, sir.
Still not.
But screen is visible. No issue. It's not in full view mode, but it is visible. Can you share the entire screen? In that there may be an
Yes.
option, share entire screen. After
Full
that
screen,
you open your, yes, sir,
Entire screen. Okay.
entire screen. Then you can project your PPT. Then in that you go to slideshow mode and full screen.
Okay.
Yes, sir. Now it is clear. You please
Is it
proceed.
now? Okay. Thank you. Thank you for giving the clues. And here is one of my friends, assistant librarian, Mr. Saravanan. He is also helping me. So I apologize for the delay. There is a glitch. Okay. Shall we go into the presentation?
Yes, sir. You please.
Okay. So I would like to share my own experience with the extraction, isolation, and screening of biologically active biochemical phytochemicals, which is very important in the day-to-day life in all aspects. So WHO itself has been, proved, and they are what I can say motivate everybody, because in one of the WHO's report, it is shown that more than 80% of rural population depends on the medicinal plants as their primary health care system. So because of that, without any side effects or any deleterious harmful effects, we can boldly take the medicinal plant for any such curative things. So what is the definition for a medicinal plant? Am I audible?
Yes, sir.
Yes, sir, very clear. And one more
Okay.
suggestion, in that there may be a hide button.
Yes,
in your
yes.
screen. You please hide it. You please touch the hide.
I will do it.
Yes, sir.
Yes.
Okay, sir.
Did
You
it.
please put it in a full view mode, please.
Yes.
Thank you.
Is
Clear,
back
sir.
now? Okay. Okay. So the medicinal plants. There are n number of plants are available in both terrestrial as well as marine. So what is the specification that describes the medicinal plant? So is there any special distinguished or characteristics for that? Yes. Based on the constitutes an effective source of both traditional as well as modern medicines. So I hope you understand the first line, because these plants are the origin for or effective source of origin for both traditional as well as medicinal modern medicine. That is where it is termed as the term coined as medicinal plants. What is its importance? Herbal medicine have been proved to have genuine utility. Why? Because as I told in my first line, without any hesitation or without thinking about any deleterious or harmful effects that may arise in future, we can take it on the whole. That is what the importance of herbal medicine is talking about. Also, we are all aware that there are different system of medicine followed in India called as Indian system of medicine, namely the Ayurvedic, Unani, Homeopathy, Siddha system of medicines. All these are what I can say is a very ancient medicine which is now being scientifically proved, and to validate those things to bring out as into the day-to-day life. That's where people are trying to do. Also, WHO is motivating the scientific research community as well as the physicians. So what is this medicinal plant is rich about? They are richest biosources for drugs of both traditional and modern medicine. Also, it is used in the field of nutraceuticals, all food supplements, pharmaceutical intermediates, and chemical entities for many of the synthetic drugs. So when we come to the phytochemicals, what is a biophytochemical? So actually, it is termed as natural bioactive compounds found in different parts of the plant. So namely, is there any specific part of the plant? No. Head to toe, like, from top to the tender leaves till the root, leaves, flowers, fruits, stem barks, and roots. Everything has different compounds that are bioactive and which can be used in the field of medicine. So which provides a definite physiological, what is the importance? Why we are more interested towards the bioactive compounds present in the plant origin? It is proven that, and also we can boldly say that, provide definite physiological action on the human body. So what is a bioactive substance? It includes different components such as tannins, alkaloids, carbohydrates, terpenoids, steroids, and flavonoids, and also a number of other compounds as well. So they are widely used in the human therapy, veterinary, agricultural, scientific research, and other countless areas. It also shown to have inhibitory effects against all type of microorganisms in vitro. So why we are specifically saying that in vitro, inhibitory effect against all type of microorganism? We all know that in one way or other, all the microorganisms, the what is called health hazardous microorganisms, which causes deleterious effects in one way or other, either as a disease or as a disorder. Okay, so if you fall into something like a stomach or what is called slight fever, viral fever, maybe due to that microorganism. There is cancer which is caused due to microorganism. HPV is one of the culprit. Human papillomavirus causes cancer. So that is what I am defining, that disease to disorder. In each and every case, these medicinal plants are effective. So come to extraction. So how we can use this phytochemicals? So on the whole, instead of using on the whole, why we have to extract it? Because we know that plants has more than one compound in its body. So the why we should concentrate on the extraction, isolation and screening? Because of the presence of n number of compounds, we have to identify which is more active against or which is more potent against a specific disease or disorder. So those compound has to be isolated, and that should be screened based on in vitro studies and then in vivo studies, and then that is a scientific validation methods. So based on screening, and then it should come into the use of human, because it's going to play a role in ultimate goal is save the life of the human. So extraction is the separation. What is the definition for extraction? Extraction is the separation of medicinally active portion in the plant tissues using selective solvents through standard procedures. So what are those? Some of the basic parameters to influence, standard procedure means some of the basic parameters that should influence the quality of an extract. Plant parts used as starting material. So solvent used for the extraction and extraction procedure. All these things has to be very, we have to give very much importance towards all these things. So next come to the choice of a solvents. So successful determination of biologically active compounds depends on the type of solvent used in the extraction procedure. Am I clear? Are you able to clear? Listen me. Property of a solvent in the plant extraction should be like the solvent should have low toxicity, ease of evaporation at low temperature, and promotion of rapid biological absorption of the extract, and also it should act as a preservative for few of the constituents. So come to the factors that affects the choice of the solvent. Quality of the, because if you miss, as I explained in the previous slide, this low toxicity should be there, ease of evaporation should be there, promotion of rapid biologic absorption should be there, preservative action should be there. So all these has to be considered while using that. So what it will happen if you are not considering, or we are not that much keen into those things? The quantity of the phytochemicals extracted may be a lot, but if you are not given those parameters is not given importance, means the quality of the product will be lost. So for that purpose, we have to select a suitable solvent which is responsible to maintain the integrity of the drug action, and rate of the extraction should be fast. At the same time, we should not lose its potency. Diversity of different inhibitory compounds should be extracted, and ease of subsequent handling of the extract. Toxicity of the solvent in the bioassay process should be minimized. Potential health hazard of the extractants can be minimized. So because of these various reasons, we have to be very much give important very much knowledge, and also while choosing the solvent we have to give very important to that. So what are the active solvents that are being used commonly to extract the different compounds? when we are focusing towards a specific group of compounds, there are different solvents. So you know that as a, most of you are chemistry based, I think so. You know that polar and non-polar compound, solvents. So based on the polar to non-polar solvent, you have to choose. So there are different solvents starting from water. In even though in the term of chemistry we are saying that polar, non-polar. So in term of biological action, we'll call it as hydrophilic and hydrophobic. So you also are very familiar with the hydrophilic and hydrophobic nature. So water to acetone, water through acetone, like water, ethanol, methanol, chloroform, ether, and acetone. Either one of the solvent can be used. Depends on the targeted compound what we are planning to isolate from the specific part of the plant. So, so these examples gives, underneath the solvent system, there are specific compounds which can be isolated, extracted by using the different solvent system. I hope you'll understand me. So next, come to the techniques. Some of the techniques used in the medicinal plant extraction. What are the techniques? Very much we have to be that are being used. So the first one is the plant tissue homogenization, then maceration, infusion, percolation, digestion, decoction, succinate extraction, which is called a hot continuous extraction, and sonication ultrasound extraction. These are the various methods that are being used in the field of extraction of active constituents from the medicinal plants. Let's discuss very quickly one by one, at the same time briefly. So what do you mean by plant tissue homogenization? As soon as you get the plant, either wet or dried. Dried means we can dry it either under the shade or sunshade also. So depends on the, again I'm saying, depends on the compound which we are focusing on the particular extract. So some of the compounds under the sunlight, some of the compound, if you dry the leaves under the sunlight, some of the compound, for example, chlorophyll is the best example, it will be dry, it will be gone away. So in that case, you have to take it inside in the sunshade, under the shade, and you have to dry it. Wet plant also can be used, either case wet or dry plant. The plant should be grinded into a blender to fine particles and along with a certain quantity of the suitable solvent. So once you grind that into fine powder with the fine particles with the solvent, shake that vigorously for 5 to 10 minutes or left for 24 hours. Either you want to do it immediately, you can shake it vigorously for 5 to 10 minutes, or we can left it for 24 hours, after which the extract can be filtrated. The filtrate can be dried under reduced pressure and redissolved in the solvent or centrifuged for clarification of the extract, and that extract can be further used for your study. Next, come to the maceration. What is a maceration? Where coarsely or whole powdered crude drug is placed into a stoppered container with the solvent and allowed to stand at room temperature for a period of at least 4 days with frequent agitation until the soluble matter gets dissolved. Then the mixture is strained and mat is pressed. That is, the solid material will be compressed into the bottom. So in that case, you can take out the liquid. The combined liquids can be clarified by filtration or decontamination after standing. Then come to the infusion. This is another method how the extraction can be efficiently done. Like fresh infusions are prepared by macerating the crude drug for a short period of time with cold or boiling water, and these are dilute solution. This is just it's called infusion. You're grinding or macerating these material with the boiling water or cold water even, then that can be filtered and used. Readily soluble material will be in the form of crude drugs. Then come to the digestion. This is another method, a form of maceration in which gentle heat is used during the process of extraction. Used when moderately elevated temperature is objectionable, when means When moderately elevated temperature is not required means gentle heat has to be given there, and then the solvent efficacy will be increased in that case to get a better yield of the constituents. Then come to the decoction. So decoction is prepared in a 3 different stages, like 3 different process. So one is the crude drug is first boiled in a specific volume of water, that is maybe 1:4 ratio for a defined time. Okay, then it should be the volume is, once the volume is reduced to, for example, if you are using 4 liters, it has to be reduced to 1/4 of that original, and cool, then strained and filtered, then suitable for extracting water soluble compounds and heat stable compounds are there, typically used in the field of Ayurvedic extract. So next is the percolation. So in the percolation method, what we are going to use, used most frequently to extract the active ingredients in the preparation of fluid extracts. So when you have the fluid extracts from the plant or plant origin, this is a suitable method to get the full active compound. When you want a residue to be prepared or powder form have to be retained back, means other methods would be better. If you want to use it in a liquid fluid form itself, percolation is the better method. The solid ingredients are moistened with an appropriate amount of specified menstruum and allowed to stand it for approximately 4 hours in a well-closed container. After a stand time, the mass is packed and the top of the percolator should be closed. The mixture is allowed to macerate in the closed percolator for a period of 24 hours, and then additional menstruum should be added if it is required until the percolate measures about 3/4. So keep this in mind, keep 3/4 of the volume and the required volume to finish the product. There are the area in this pressed and then expressed liquid is added to the percolate. So sufficient menstruum is added to produce the required volume. The mixture is finally clarified by filtration or by standing, followed by decanting. Then it can be used in the form of liquid itself, that is a fluid extract. Then come to the oxalate extraction. So this is otherwise called as hot continuous extraction. So what is the oxalate? In most of our laboratory experiments, we are using the oxalate extraction method. So how we are doing this? So maybe some of you are familiar with this method. So the finely ground crude drug should be placed into a porous bag or thimble, which is made up of a strong filter paper, that should be placed inside the oxalate apparatus. The extracting solvent in the flask is heated under regulated temperature, and the vapors passes through the solid material, condenses into the condenser. The condensed extractant drifts again into the dimple containing the crude drug. Again and again it will come like that. So when the level of the liquid in the chamber rises to the bottom of the siphon tube, the liquid contents of the chamber siphon into the flask, and the liquid again condensed into the chamber. Sorry for the typo. Condensed to the chamber siphon into the flask. This process is continuous and is carried out until a drop of solvent from the siphon tube does not leave a residue when evaporated. Okay, so in this way we have to collect the extract and then dried under controlled temperature and pressure, evaporated. Then the residue can be further taken for its use. So the next method, which is called sonication. So this is not that much appreciable method. I will explain why it is not that much appreciable method. But even though, for example, when preparing the what is called the nanoparticle kind of things, this method is the more suitable method. So which involves the use of ultrasound with frequency of ranging from 20 kilohertz to 2000 kilohertz, depends on the particle size. It is required high current when ultrasound has to be applied to increase. Why? Because it's to increase the permeability of the cell wall and produces cavitation. Okay, so as I told, this is not that much, advisable method in the preparation of the natural product drugs. Why? Because this deleterious effect come out from the ultrasound. As I told, somewhere between 20 kilohertz to 2000 kilohertz of sound waves has to be applied. So here it is a proven thing that anywhere greater than 20 kilohertz of the sound waves is deleterious on the active compounds of the medicinal plants through formation of free radicals and consequently undesirable changes in the drug molecules. So as we all know, so what is the free radical? Why we are concerned about the free radicals? Free radicals is is because of the loss of electron, it is being formed. So those free radicals will be always searching for a binding molecule that hits the tissue. So here we are talking about the drug. So it will attach with the replaceable atoms present in the drug, and it will stay there. Once you are taking that as a drug, again the free radical will be released into the physiological system and made consequent effect into the human system. So free radicals are the main culprit for most of the disorders these days. Then come to the effect of extracted plant phytochemicals depends on the nature and the origin of the plant material, where it has been collected, and under what degree of what degree of the importance we have given for its processing, moisture content of the plant, and also the particle size which is present. These are which is prepared. These are all going to give the efficacy, improve the efficacy of the extracted plant phytochemicals. Then come to variation in the extraction method. So what is the variation in the extraction method? Length of the extraction period may be varied, depends on the compound what we are targeting, our focus compound. And then the solvent, as we have discussed in the earlier slides, like the solvent choice of the solvent should be very much important. So, and also the pH of the solvent what we are going to use depends on whatever compound, type of compound. We have to also be very keen in that to pH of the solvent. On temperature, we know that in boiling temperature, something above 60 degrees centigrade or 70 degrees centigrade, most of the compounds will lose their original property and it will be converted into other form. If you want really the native function, native originality of the compound, we have to be very much, we have to give very much importance in the temperature also. And also the particle size of the plant tissue and sample to solvent to sample ratio. In one of the extraction method, what is called the boiling method, we discussed about 1:4 ratio like thing. That solvent to sample ratio is also very much important. So once you extract those compounds, so how we are going to, because you know better than me, I think so, doing the qualitative analysis and quantitative analysis of the phytochemical compounds, how we can be assessed what is the functional property of those compounds present in the particular extract. There are various test methods available. For example, for the alkaloids, drag and drops test, Wagner's test, and Mayer's test. These are the tests which is being used to confirm the presence of the alkaloids, and flavonoids through the ammonia test, sodium hydroxide test, and tannins by using the ferric acid, ferrichloric test, and the phenolic compound gelatin core test, and the saponins by using the lead acetate test and foam test. All these tests can be used for the qualitative analysis. And when you come to the quantitative analysis, in continuation of the qualitative analysis itself, we have also terpenoids which can be analyzed using the sulfoxylase test, carbohydrates by using the Molish test or Felling's test, proteins by using the Poirot test, glycosides, Legal's test, or Keller's killing test. When you come to the quantitative analysis, whatever for the only to say for the sake of knowing the present, either it is present or not, we can use the qualitative test. So that quantitative test is very much quality to check whether it is present or not. At the same time, quantity also, because we are making much effort to take out the compound by collecting the material. So there we have to be very careful in quantifying the compounds also. For example, this total phenolic compounds by using the Folin-Shio-Calcium method, and total flavonoids can be used by using the calorimetric method determined by Yoon et al. So these are the methods to use the quantitative analysis, which is available in the published literatures, where we can dig in and find out the different methods. If you have questions, you are always welcome as well. And coming to the next slide, what is this? These are some of the commonly used product in our day-to-day life, which also rich in most of the active compounds. So for example, if you go with this what is called that turmeric, which is rich in curcumin, has been proved in most of the disease and disorders. It's very beneficial. Then Osmum santum, this is one of the sacred plant and holistic plant. This is also rich in the compound eugenol, which is a potent anti-cancer compound, which I'm also going to share my main focus here today is going to be screening of biological action of the compound, where I used this plant. That is where I was interested in Osmum santum. I used it in my study. I will show my results later. And then Bacoba moneri, which gives glucosides, which is very potent against the memory loss and all the neuron related studies. And Vidania somifera, this is also called as, in Tamil we call it as Amukra. So this Vidafera A is an active compound, and also vithinosolides are also present in there. So these are anti-inflammatory as well as anti-cancerous drug. So when you come to Azadirachta indica, this is what I can say. It is one of the very important tree everywhere we can see, which has the nimbin, neem tree. It is nothing but neem tree. So we all know the importance of the neem tree, starting from basic some viruses, smallpox virus, chickenpox virus, everything is being controlled by this compound. Also, it has potent anti-cancer effect. And come to the mentha species, that is this is also we are using in our day-to-day life, which is the example is the pudina leaf, so which has the menthol. And also ginseng root is called ginsenoside. This is a potent anti-inflammatory as well as anti-cancerous property compound. So, as I have told you a few seconds ago, so screening of an ethanolic extract of the dulce for its apoptotic induction potential against prostate cancer cell. This is one of my hands-on experience, where I have extracted the compound by using the Soxhlet apparatus and take out the extract. So I used it in my study to prove that it has Osmum santum, because it is a rich source of eugenol. Eugenol is a potent anti-cancer drug. Based on the literature search, I found out that this compound is abundantly present in this plant. So this plant can be used in the study, and the paper has been published in Journal of Medicinal Food in 2-3 years before, I think. So before I go into the results of the study, what I have did, so I'll give you a brief brief outline about what is a breast cancer, what is a cancer, and how it is going to be, what is the apoptosis, and how it is inducing the cancer cell death. So cancer, we all know that abnormal growth of any tissue or uncontrolled multiplication of the tissue or cells formed into a lump of tissue is called cancer. So those cancer is being broadly distinguished into two types. One is benign tumor, the other one is the malignant tumor. So benign tumor, of course, even though it is tumor, it is a little bit harmless when compared to malignant tumor. Why? Because the benign tumor, or otherwise called as the local tumor, which will not spread out into the neighboring tissue or to the other part of the body. But the malignant cancer is the culprit, which will spread out with the own or origin of the development as well as to the other parts of the body. So here, coming to the prostate cancer is the male. Prostate is a gland which is a male specific gland. It is only for male. So male specific gland, which is very dangerous. There in United States of America, this is one of the most prevalent cancer in male, and in female it is a breast cancer. Compared to in India, it is breast cancer and ovarian cancer for the female, and this lung cancer used to be the prevalent cancer, but nowadays the prevalence is going towards the oral cancer, where the tongue and the head and neck area is being involved. So what is the role of, what is the meaning for the apoptotic induction? So what is apoptosis? Apoptosis is, that a cell is being growing and dying every day, each and every minute. So that is the cell turnover is called apoptosis. Apoptosis and the cell turnover is being regulated in a normal way or balanced way, means then the cell, what is called the lump of tissues will not accumulate anywhere, thereby there won't be any cancer. So apoptotic potential has to be increased. Why? Whenever the cell death is decreased and uncontrolled multiplication of the tissue is being happen, then we have to balance that by increase the rate of cell death, thereby balancing the cell growth as well as the cell death. So that's where I used the extract collected from the Tulsi plant and used to see its potency against the cancer cell. So this is where I was telling that biological screening of the phytochemical. Okay, so let us come to, this is the cancer cells which I have grown as layer. It is an in vitro study. So I have grown it as layers into the flask. So figure 1A and 2A are the control. So the panel 1 is talking about 24 hours of treatment with the different concentration of the extract which I have prepared from the Tulsi by employing the oxalate extraction method, and evaporated the solvent and use the residue with the suitable solvent to mix into the growth medium. And the panel B shows with the different concentration for 48 hours of incubation period. So where comparatively, when you see the 2C or 2D, which gives more cell death, because there are, you see, I don't know whether you are able to see my arrow, so pointer. So this pointer showing that these are the dead cells which are accumulated, and these are the empty area. Just compare this first flask and the third or fourth flask, so you are able to, you can see the difference. So the same result has been counted, number of cells has been counted and plotted into a graph to find out the effective dose, which we called ED50 or LD50, lethal dose 50. So where we can have we can achieve 50% of that cell death in a desired time period. So the bottom two lines are the prostate cancer cells, and the top two lines are the normal cell line. So why I use the normal cell line? To show that our drug is not potent, not toxic to the normal cell line. It selectively kills the cancer cells, not with this. So this is the uniqueness with the what is called the natural product system or herbal medicine system. Say for example, it's not a blaming. At the same time, I want to explain you about the a deleterious effect compared to the, non-deleterious effect or beneficial effect of the herbal product compared to the Western medicine. So Western medicine, for example, if you are going to a doctor, to discuss about you have fever or cold or sore throat something, so they'll give some paracetamol. Along with that, they will also give some supplement. What is that supplement? It's a vitamin supplement they will give. So just think about all the vitamins, even though it is being synthesized chemically, so those vitamins are naturally available in the different parts of the plants and plant products. You all know that. So B complex or A or B complex, for example, B complex, which is a rich source of the B complex vitamin, is our normal starch water, rice polish is the best source of vitamin B complex. So folic acid, niacin, everything is present there. So these days, people are going back to the ancient days, getting the rice bran oil and what is called the starch water. That I think most of you are from Tamil Nadu. You may understand this word, Niragaram. You know what is the cost of Niragaram in USA? It is $7 a liter. So it is unbelievable. So okay, let's come to our topic. So this normal cells was not found to be dead by using our drug extract. At the same time, the cancer cells at a different time point, the bottom one is of 48 hours and the top dash line is the 24 hours. So comparatively, those shows, cell death, very high cell death was observed. So coming to the next proof for that. So this is one of the how the cell death was caused by arresting the cell growth, at the same time, killing the existing cell. Killing the existing cell means, so we have to stop the energy supplement to the cell. Energy, as you all know, what is the powerhouse of the cell? Where the energy is being produced? Mitochondria, cell mitochondria. So when you disturb the mitochondrial architecture, then the energy will not be produced. As I told, our drug is very specific to the cancer cells alone. In the previous result, I showed that normal cells have not been killed, but at the same time, the cancer cells has been highly killed. So how it killed? It disturbs the mitochondria, thereby stops the generation of the energy for it. So we all know why we are eating. Our food is being digested, and those particles in the form of NADPH and NADH is being generated in the mitochondria through the various biochemical metabolic pathways. It is being generated into energy. By that way, we are getting energy, okay, in the form of electron. So if you stop the formation of those energy electrons, then what it will happen? So automatically, the cells will be dying. Cells will be dying. So for that reason, I was showing that the mitochondria is the major focus. Disturbance of the mitochondria is the major focus of our drug, whatever we used in our study. So in the first panel is the one which shows without any disturbance, because this is not treated with any drug. The red fluorescence shows the healthy mitochondria, where we are not able to see any disturbance in the green fluorescence. I'll explain you what is the green fluorescence. So where we are not able to see any green fluorescence. So while merging those two, green and red fluorescence, in the last panel, we are able to see only the red fluorescence alone. At the same time, when the cells were treated with the different concentration of the drug, so it shows the healthy mitochondria, less number of healthy mitochondria, and the green fluorescence are called the disturbed mitochondria, cells that carries the disturbed mitochondria, which is about to go into the cell death. So while you merge these together, so you see, you can if you are able to see my pointer, please look into these two dots. These are the cells which got disturbed and which is obviously shown by the green fluorescence. When you merge those together, the green on top of the yellow, red mitochondria gives a yellow fluorescence, kind of yellow fluorescence. So this shows the induction of that is called apoptosis, programmed cell death, stage by stage. It is not killing directly like dies the cell or something. First, it will enter into the mitochondria, and it will stop the energy production. So energy production is stopped means automatically the cell will starve for its energy for survival. So while starving, it is going to be dying automatically. So that is what we confirm. This active component present in the eugenol present in the dulce induces apoptotic mediated cell death. That is, programmed cell death was initiated. So the same was also confirmed using the what is called flow cytometric analysis, which is called a fluorescent assay with the sorting cell sorting method. So here this is having 97% of the cell, live cell, which was not treated with any of the drug, and B was treated with 50%, 50 micrograms of the drug for 12 hours. Because we want to catch the early stage of the cell death, we used very minimum quantity, and time period was also less here, where we are able to see comparatively this healthy cells were 97. Here we are able to achieve like 5% of the cell death with 50% and 13% of cell death with 100 micrograms. And comparative, for comparison, we used one of the other compound called topotecan, which causes 28% of the cell death. Even though that is a synthetic compound, our compound is a natural product with less side effects. Further, what is the next consequence? If the programmed cell death is being initiated, what is the before we discuss about this DNA fragmentation, I would like to show the induction of the program. What are the proteins mainly involved into the cell multiplication and cell death? So these are the two main proteins. One is PARP, the other one is polyadenylate ribosylate. The other one is the BCL2. BCL2 is a pro-apoptotic protein, which is anti-apoptotic protein, which increases the cell number. The cleaved PARP is the one which initiates the cell death. So here from the initially the PARP will be not cleaved in the cancer cell because it is actively involved into the cell multiplication. Also the BCL2. So when you use the drug concentration of different concentration of the drug like 25, 50 and 100 micrograms into the flask for treatment, slowly it is being increased. The cleavage the cleavage is being increased. At the same time, BCL2 is found to be decreased. So with the high concentration, more of the cleavage is was observed. Also, the BCL was completely compressed. So these proteins shows that the compression and the cleavage of these two proteins shows that the cell death is being increased. Not only cell death, by examining this PARP cleavage, we are able to distinguish, show that cell death is being increased. BCL2, as I told, it is a major protein involved into the cell growth or multiplication. This BCL2 decrease on the use of the drug shows that the cell multiplication is also stopped. So further it was confirmed, multiplication, how it is happening? Through the nucleic acid. What is called replication of the nucleic acid present in the cells. So cell multiplication, one cell is being copied into the 2 to 4, 4 to 8, 8 to 16, like 2 to the power n times. So when the whole nucleic acid present inside in the nucleus of the cell is being copied or multiplied, that gives the copy of the cell. So when these two proteins are being disturbed, there is no way to multiply or copy the nucleic acid as itself. Why? Because so this PARP cleavage, as I told you, adenosine ribosylate phosphate. So phosphatase. So phosphatase function is it cleaves the phosphate backbone of the nucleic acid. So if the phosphate backbone of the nucleic acid is being cleaved, especially at the adenosine site, okay? So especially at the phosphate backbone is being cleaved by this particular phosphate means it is going to be broken and pieces of nucleic acids are going to be there. It is not a full copy of the DNA. So full copy of the DNA is not there means it's called DNA fragmented. That fragmented DNA will not mimic the action of the cancer cell. That is where it is not able to act as a exact progenitor of the cancer cell. So now, in our study, we also confirmed through the electrophoresis method that Dulce extract at its different concentration was able to induce the DNA fragments. So this is the loaded DNA with don't have any lane 2. Just if you're not able to see my arrow, just see into the lane 2 which don't have any drug at all. Lane 3 has the cell was incubated with 25 micrograms of the drug for 48 hours, and this lane 4 was 550 micrograms for 48 hours, and lane 5 is for 100 micrograms for 48 hours. So obviously in lane 2 and lane 1 was the ladder marker to see at which area these fragments are being observed. So in lane 1, lane 2 and 3, lane 1, 2 is very clear, there is no fragments at all. Lane 3, we are able to see one fragment near the 500s base pair, and in lane 3, 4 and 5, it is obvious that lot of fragments were present in between this 1800 to 380 base pairs length. So this shows that the the concentration of the Dulce extract which we have used in the study has proved that DNA fragmentation is also happened. So as I told, the programmed cell death is being happened by using the eugenol containing, Dulce extract which contains the eugenol. Okay, so in this way, the stage by stage killing, or what I can say, it's not directly hitting the cell to destruct by itself, but initially, if you see into the first result, it is entering into the mitochondria, stopping the energy production. So once energy production is stopped, what is happening? This apoptosis is being initiated. So once it is initiated, these proteins are being changed. So which is important for the cell growth is being completely compressed, which is important for the lysis of the DNA is being obtained. The fragmented part is being there. So then it causes the DNA fragmentation as its final product. So this shows a very clear, it's my hands-on experience on screening one of the natural products which I have extracted from one of the plant and proven that it has a potent effect against the prostate cancer cells. So this is one of the examples for the screening of phytochemicals for its biological activity. So in this way, we can also compare lot of other biological activities by using the different screening methods which are available in the published literature. So this I think this ends my I won't say it's ends my, for now this much is enough, I think so. If anybody has any questions, open to discussion now. Hello?
Yes, sir. Audible.
Is it audible?
Yes, sir. Yes.
Okay. Yeah. I think we can have, I dragged the time till 4:13, I think. I'm sorry for the late. At the same time, we can have one or two, discussion or question if anybody has.
If you have any queries.
First of all, was it clear? Audience, I would like to know your, intake, like how my presentation received to you. Are you all able to understand my presentation?
Audience, anyone can reply, please.
Oh, but sorry, everybody is under the mute mode, so I could not hear anybody.
good
That's
evening,
fine.
Yes,
Of
sir,
course.
you're
evening,
very clear on all your particular
Good
explanations.
evening, sir.
Some questions.
Sorry to disturb you.
I'm Gunavati, sir. I have a question.
Okay, ma'am. Okay.
Yes, madam,
I have
please.
some questions.
Could you tell us some of the assays that can be done for cardiac cells? You have explained this for the cancer cells, right, sir?
Yes,
I
yes,
need
yes.
some assays for cardiac cells.
Yes, we can also do that. Rat astrocytes are available, in the cell culture. I mean, there is one in the Lucknow, no? This tube culture place where we can get the rat cardiocytes. Cardiomyocytes can be obtained. The same methodology can be applied. We can culture that into the medium, and we can use the any of the active components and see that.
Yes, sir. Thank you so much, sir.
If you need further help, always I'll share my email ID. My friend Anand, Dr. Anand is there. You can get my phone number or email ID. I can also very much, happy to help you to get more information if you want.
Thank you, sir.
You're welcome.
Participants, if you have any queries, kindly clarify with us.
Sir, you please look at the chat box. One of the professors, Dukaram, I think, share certain things.
Yes, Shivaji Arts, okay. Send paper to email ID. I'm at, okay. Research in chemistry volume publication. Okay. So he's asking which paper, advanced researches in there. Okay.
So can we conclude, sir, then?
Sir, Sivaraman sir is, I think, interfered and he may...
Sir, good evening, sir.
Yes, sir.
Yes.
Good evening, sir. Good evening, sir.
This is Dr. Sriraman from Chemistry Department, sir.
Hello,
So,
sir. Nice to hear you. I'm not able to see. Nice to meet you. Nice to hear you.
It was a nice presentation, sir. And
Thank
I
you,
have
sir.
few questions in the characterization part. Can I ask you, sir?
Yes, sir. Please.
Some of the natural products contain the glucose particle or sugar particles, carbohydrates.
Yes, sir. Yes, sir.
So while doing GCMS, which instrumentation will be fine for the detection of the mass, sir?
Up to my knowledge, I'm a basically biochemist. I can share my knowledge. Up to my knowledge, it's better to go with GC. I think so.
Okay, sir.
Yes.
Okay,
I can
sir. Thank you,
also, I can also refer something else if you want. I can, send it to you.
yes, sir. We will contact you. Keep in touch with you.
Sure, sir. Sure, sir. We'll be in
Thank
touch.
you.
Also, you can use the HPLC method. Okay. Anybody else?
Sir, I had one layman question.
Please, sir.
Good evening, sir. Sir, nowadays
Yes.
in COVID pandemic period, everyone suggesting so many traditional medicines like Abasura Kodinir and other medicinal value plant extracts. Are they
Yes,
really control the viruses?
Sir, it is. It's like nobody knows the final determinant of the virus. What is the structure, and it is still under validation only. So assumption by having this and that, we are assuming that more than one by eating more than one compound, by having one more compounds, our immunomodulation can be happen very faster. So immune system will be boosted. So when immune system is boosted, then we are ready to fight against the virus. That is the basic concept, sir. So that's a general thing only. It is not any proven thing that any of these things is being virulent, toxic against that particular virus. It has not yet been proved, sir.
Okay, sir. Thank you, sir.
You're welcome.
Any other questions from participant side and from our faculty side?
Participants, if you have any queries, kindly clarify with our speaker.
Okay, sir. I think no any queries.
Okay. So in that case, I think I would like to share a word of thanks to everybody. Like the management who give this opportunity for me to be with you, even though online. So the management who permit us to meet us online to share my experience and knowledge on the field of extraction, isolation, screening of biologically active phytochemicals. Also the respected secretary of Srimad Andhayuthapani College, director of the Srimad Andhayuthapani College of Arts and Science, the principal, the vice principal, and all the dean of different faculty and all fellow members and my lovable students or participants who were there sparing their time for more than an hour. I would express my sincere thanks to everybody. Thanks one and all.
Thank you, sir.
Thank you.
Thank you, sir, for the nice presentation and for the informative and stimulating presentation, sir. It was really an eye-opening session. I hope it was particularly appropriate and helpful to all our participants, sir.
Definitely. Yeah, I can share my email. As I told, anybody can contact me at any time. I can, I'm ready to spare my hands to research. Dr. Anand knows that I started my career as a lecturer like 20 years before. After that, I quit my lecturer job and enter into the research field.
Sure.
Thank you, sir. And we have come to an end of this session. Saying thank you is the finest approach of serving as the eternal. I now kindly request Mrs. N. Kalai Selvi, Assistant Professor, Department of Chemistry, to render the vote of thanks. Kalai Selvi, ma'am.
She left. I think she had
that's
a
fine.
network issue.
Okay.
Just now I saw that
Not an
she
issue.
just a minute, sir. We will wait for a moment.
I'll carry with that, sir.
Yes.
Yes, ma'am, please.
Good evening, everyone. I consider it a great privilege to propose a vote of thanks on the successful industrial skill enrichment program. At the outset, I register my profuse thanks to our chief guest, Dr. Sivanesan Dhandayuthapani, Deputy Dean Research, Santosh, Dean to the University Ghaziabad, Delhi NCR, for his excellent talk on this topic. My enormous thanks and respect to you, sir.
You're welcome, madam.
I take this opportunity to express our sincere thanks to our secretary, director, principal, vice principal, deans for their support. I express my deep gratitude to our beloved dean, Dr. Madhu, and our HOD, Dr. Anandan, for their moral support and guidance to us in all our activities, ensure that we always do the right things in the right way. So I'm happy to express my thanks to all of our staff members from the Department of Chemistry. I extend my thanks to our participants for for your gracious presence today. Let me conclude this vote of thanks by thanking Almighty God, who has given us the strength and resolve to be here today. Thank you all. Thank you, sir. Thank you so much.
Thank you. Thank you.
So thank you, sir. Thank you so much for your kind consent and presence.
No problem, sir. You're always welcome. I'm happy to see all of you.
So kind of you, sir.
Okay, sir. I think I can end up the session.
Okay,
Huh?
sir. Okay, sir. Okay, sir. Thank you.
Okay. Catch you later.
I'll
Thank you so much.
call you, sir. Thank you, sir.